Microbes are used in commercial and industrial processes for production of various organic acids. In normal conditions, these organic acids can be formed in a synthetic way as well, but nature has its own way of, of production of these organic acids. So we can rely on certain microorganisms for the production of organic acids in a biological system, that is a microbe's body. One of the important kind of organic acid which is produced by microbe is acetic acid, and the source is a bacterium named acetobacter aceti. In the production of this acetic acid, first of all we require barley grains, that is any kind of cereal or grain is required, which has to be soaked in water for 2 to 3 days. After soaking in water for 2 to 3 days, the seeds should be left in that condition for again incoming 2 to 3 days, or and during this time, when the seeds are taken out from the rinsed container, and they are allowed to dry up, at that time the germination starts. While the germination has started taking place in the seeds, during that time the seeds are crushed, and after the crushing of the seeds, this process of crushing the germinating seeds is known as malting. You should be aware that gibberellin concentration is very high, and during this situation of the seed germination, the starch content is in the highest level. All the stored starch is now on the form of getting activated and is in an active form. So as soon as the seeds are crushed, it is this crushing of germinating seeds is known as malting. After malting, water is added to these dried grains at around 60 to 65 degrees centigrade temperature, and it is allowed to stand in the same container. After um, pe- period of 3 to 4 hours, there will be digestion of starch which will be initiated, and during this time the wort formation begins. That means the sugary solution is formed because the grains were crushed, water is added to it, there is temperature, and at that temperature the solution along with the grain is boiling. So what is bubbly substance which is formed, and that is to be filtered out. Now during this situation, addition of yeast, that is Saccharomyces cerevisiae, is done, and this helps in conversion of maltose which is already uh, digested by the or which has already started getting digested by the high temperature, will get converted completely into glucose, and then glucose is converted into alcohol or carbon dioxide, alcohol and carbon dioxide. Finally, after fermentation for around 10 to 15 days, beer is formed, and it is an alcoholic beverage. Now from that glucose which was formed, vinegar is extracted. Now here the interesting part is that malted part from where glucose was formed. After the formation of glucose, alcohol, or after the formation of ethyl alcohol, that ethyl alcohol is converted by acetobacter aceti into acetic acid due to further fermentation. And the usage is in form of vinegar, and sometimes it is used for manufacturing of acetate rayon. Plastic production is also done with the same. Rubber production and photographic chemicals are, for the photographic films we need various chemicals. Those chemicals are also formed with the help of the same process. So this is the use of acetic acid. Next is about the butyric acid. It is produced by action of bacteria, and the bacterium is a gram positive anaerobe which is found in the intestine of human beings. And sometimes it can be used as a prebiotic agent for curing various diseases. So the main source for this butyric acid is Clostridium butyricum, and it helps in rancidity of the butter, or at times it helps in clinical treatment of inflammatory bowel disease or antimicrobial associated diarrhea. These diseases can be cured with the help of Clostridium butyricum. Citric acid is formed by from the fermentation of sugary syrups, and it is fermented with the help of Aspergillus niger and Penicillium. Both, it's not Penicillium, it's Mucor. So Aspergillus niger and Mucor. Uh, I just said, I just wanted to say that both of them are fungi, and fungi help in conversion of citric acid or formation of citric acid from the same fermented syrups. So, food preservatives or flavoring agents are used with the help of this. Certain vitamin C containing tablets are made, and ink and dye production is also done with the same. Because if you remember, aspergillus has a typical purple color, and that can be used for making dyes. Gluconic acid is formed from or is obtained from Aspergillus niger and Penicillium. The main purpose or use of gluconic acid is to provide calcium for the cows or for babies or even the lactating mothers. So it is helping in calcium content or increasing the calcium content of any individual. One more important thing is gluconic acid is formed by addition of uh, COOH group, that is carboxylic group, to the glucose structure. So just one small conversion occurs for the production of this position 1, which used to contain just H, now contains COOH in case of the structure of glucose. Next kind of organic acid is lactic acid, and it is produced by the fermentation of sugars. And as you can see, Streptococcus lactis and Lactobacillus are kind of bacteria, whereas Rhizopus is a kind of fungi. So both fungi as well as bacterium help in production of lactic acid. It is used in food products like pickles, canned food products, bakery items, in making dyeing or tanning products. So this form of lactic acid, which is used, is an organic way of getting very high quantity of lactic acid from certain bacterium, and it is helpful in industrial or commercial purpose of bakery as well as industries. So this is about the use of organic acids in or production of micro or use of microorganisms in production of organic acids.